Imagine this huge natural pharmacy, just packed with compounds, hundreds of them. But we've really only properly explored maybe two. THC and CBD, the famous ones.
Right, the ones everyone knows.
Exactly. But what if the real breakthroughs, the keys to new health stuff, are hiding right there among the other 120 plus compounds in cannabis that we just haven't looked at closely?
It's a really compelling thought. It speaks volumes about the complexity and, well, the potential just sitting inside this one plant, a huge underexplored landscape, like you said.
Absolutely. And that's really our mission here on the Deep Dive. We take the sources you give us, unpack the key insights, and give you that shortcut to being genuinely well-informed. So today we're diving into this fascinating world of minor cannabinoids. What are they? Where do they come from? And what's this surprising potential they might hold for, our health and well-being?
Precisely. And when we call them minor, it mostly reflects how little they've been studied biologically in the literature. It doesn't always mean they're only in tiny amounts. Oh.
Okay,
Yeah.
many are found in smaller concentrations, sure, but the main reason for that minor label is often just, we haven't researched them as much as THC and CBD.
That makes a lot of sense.
Right.
We hear about the big four phytocannabinoids, right? 9-THC, CBD, CBG, and CBC.
Yeah.
But you're saying even some of those aren't actually major players in the plant itself, concentration-wise, even though they get lumped in with the big guns.
That's exactly right. Take cannabichromene, CBC. It was initially overlooked, sometimes even confused with CBD. And while it got grouped into that big four early on because of, isolation and some research, its actual concentration in the plant is usually much lower than THC or CBD, sometimes way lower, like 2 orders of magnitude.
Wow.
And here's something crucial. The forms we usually talk about, the active ones like THC and CBD, they actually start out differently in the plant. They begin as acidic precursors.
Acidic precursors,
Yeah.
like not quite ready to go yet.
Exactly.
Yeah.
The plant naturally makes these acidic forms like THCA and CBDA. Those are the native cannabinoids, really.
It's only through a process called decarboxylation, usually triggered by heat, like smoking or cooking or even just light and time, that they convert. They lose that acidic part and become the neutral, active forms we know better, like THC and CBD. So what's actually in the living plant is often different from what ends up being consumed.
Okay, that's a really fundamental piece of the puzzle.
Okay.
And if we're talking origins in the plant, we absolutely have to mention the mother cannabinoid, don't we? Cannabidiol acid, CBGA.
Indeed. CBGA is the foundation, the parent molecule. Think of it like the main trunk of the cannabinoid family tree inside the plant.
Okay.
It all starts with its biosynthesis. A molecule called geranyl pyrophosphate combines with olivetolic acid. That makes CBGA.
Got it.
And then from that central CBGA stem, different specific enzymes act like pathway directors. They guide the synthesis to create those various acidic forms, THCA, CBDA, CBCA, and others.
And then those turn into the neutral forms we know.
Exactly, through that decarboxylation we talked about.
So CBGA is the starting block for pretty much all the classic cannabinoids.
Yeah.
But what about those veranoids you touched on? Things like THCV and CBDV, where do they fit in?
The veranoids, yeah. They're a really interesting subgroup. They actually come from a slightly different but related mother molecule, cannabidiol acid, or CBGVA.
Okay, CBGVA.
Right. The key difference with veranoids is their structure. They have a shorter side chain, a 3-carbon propyl chain, instead of the more common 5-carbon pentyl chain you see on THC or CBD.
So a small structural tweak makes a whole different family.
Pretty much. It's amazing how the plant uses just a few basic building blocks to create this incredible variety of compounds, each with potentially unique effects.
Yeah, it really is like nature's own incredibly sophisticated chemistry lab, isn't it?
Absolutely.
And that diversity is exactly what we're going to explore next. We'll dive into some specific examples of these minor cannabinoids. We'll focus on what the research suggests about their potential, maybe for neurological issues, inflammation, metabolic health, things like that. Let's start with cannabidiol CBG.
Sounds good.
Okay, let's kick off this deep dive into the specifics with CBG. You hear it called the mother sometimes, but as we just established, it's actually a direct descendant of CBGA, right?
Correct. It comes from the decarboxylation of CBGA.
So what's its unique pharmacological fingerprint? How does it interact with our bodies?
Well, CBG is pretty versatile. It's known as a weak partial agonist at both CB1 and CB2 receptors.
Okay, break that down. Agonist means it activates the receptor.
Right, like a key turning a lock. Partial means it only turns it part way, so the effect isn't as strong as a full agonist, like THC at CB1.
Got it. Weak partial agonist.
But what's really interesting about CBG is its broader reach. It interacts with a whole range of these things called transient receptor potential channels, TRP channels.
TRP channels, those are like cell sensors.
Exactly. Tiny molecular sensors on our cells detecting things like heat, pain, chemicals. CBG specifically interacts with TRPV1 through 4 and also TRPA1.
So it's not just hitting the cannabinoid system, it's like it's influencing the body's sensory network too. That sounds multifaceted.
It is, and it also acts as an antagonist at the 5-HT1A receptor.
Antagonist that blocks the receptor.
Yeah, it fits the lock but doesn't turn it and stops other keys from getting in. The 5-HT1A receptor is involved in things like mood and anxiety. So this diverse activity suggests CBG isn't just working through one single pathway. It's more like it's tweaking several dials at once. That could lead to more nuanced effects, maybe even safer effects when influencing complex body systems.
That's a really powerful insight. Okay, let's shift to its therapeutic potential. You mentioned metabolic health earlier. What are we seeing there?
Well, in preclinical studies, so animal studies, mostly CBG has shown some real promise as an appetite stimulant.
Oh, interesting.
Yeah, it was observed to induce hyperphagia. That just means a significant increase in eating in rats.
Okay.
So this positions it as a potential non-psychoactive option for conditions where appetite loss is a major problem, like cachexia. That's the severe muscle wasting and weight loss you sometimes see in cancer or HIV patients.
Right.
The key thing here is it might help people struggling with appetite without the high you get from THC.
That could be a huge benefit. What about inflammation?
Also promising. Studies suggest it can help suppress the growth of certain cancer cells, for instance, in mouse models of colon cancer and some leukemia cell lines.
Wow.
It also shows general anti-inflammatory properties, and quite remarkably, it has antibacterial activity, even against tough antibiotic-resistant strains like MRSA.
MRSA, really?
Yeah. Which definitely opens up questions about its potential role in fighting infections where our usual antibiotics aren't working anymore.
So it's got some serious muscle against inflammation, maybe even superbugs. Now, what about neurological effects, specifically anxiety? You mentioned the research seems a bit mixed.
It is a bit mixed, yeah. Some studies did suggest anxiolytic-like properties, anxiety-reducing, basically.
Like how?
Well, for example, in certain tests, rats given CBG spent more time in the open central area of an enclosure, which usually indicates they're less anxious. However, other reviews looking at multiple studies concluded that CBG didn't consistently show these effects, either in stressed or unstressed rats. And they found no significant antidepressant effects or impact on fear memory.
So why the difference?
It could be down to lots of things, different doses, different models, maybe how CBG interacts with various molecular targets in complex ways we don't fully grasp yet. It just highlights that we need more focused research to really nail down its effects on anxiety.
Right, so promising hints, but still a complex picture for anxiety. Okay, let's move on to another interesting one, cannabichromene, or CBC. It's been called the abundant enigma. Why the enigma, and what do we know about how it works?
Well, it's an enigma partly because even though it can be one of the more abundant minor cannabinoids in certain cannabis varieties, it received way less scientific attention compared to THC or CBD. Pharmacologically, we know CBC acts as a partial agonist at the CB2 receptor. Remember, CBG was weak at both. CBC leans towards CB2.
Okay, partial CD2 agonist.
And it also influences our own endocannabinoid system by suppressing an enzyme called MAGL.
Okay, hang on. MAGL, can you give us the quick version? What is that? And what does suppressing it actually do?
Good question. So MBL monoacylglycerol lipase, it's basically part of the cleanup crew for our body's own natural cannabinoids, specifically one called 2-AG, but it also affects anandamide indirectly, our bliss molecule.
Right.
By suppressing AGL, CBC effectively slows down that cleanup process. It lets our natural endocannabinoids hang around for longer and exert their effects more strongly.
Ah, I see. So it kind of boosts our internal system.
Exactly. Could indirectly help with the body's natural pain or mood regulation. What's also interesting is its activity as an agonist on TRPA1 channels.
Those sensory channels again?
Yep, the same ones we talked about with CBG. Right. They're heavily involved in signaling pain and inflammation.
So CBC is hitting our internal cannabinoid system in these broader sensory pathways. How does that translate therapeutically, particularly for inflammation?
For inflammation, CBC has shown some pretty significant anti-inflammatory effects in studies. It can reduce the secretion of IL-1, that's a key pro-inflammatory signal molecule.
Okay.
And it can also downregulate a complex signaling pathway known as IL-6TYK2-STAT3. Now, that pathway is implicated in those severe runaway inflammatory responses, cytokine storms, where the immune system goes haywire.
So potential against really serious inflammation.
The potential is there based on these mechanisms. And like CBG, CBC also shows antibacterial effects against MRSA, plus some anti-tumor properties like increasing anandamide levels in certain contexts and inhibiting the migration of some types of cancer cells in lab studies.
That sounds like a really powerful compound. What about neurologically? Anything standing out?
Yes, in preclinical models of depression, CBC demonstrated antidepressant-like action. It significantly reduced immobility time in standard tests, which is a key behavioral marker used in depression research.
Interesting. So potential for mood disorders, too.
It suggests it could be a valuable avenue for future research in that area, definitely.
Any other potential uses for CBC we should touch on?
Yeah, just briefly, there's evidence it plays a role in regulating lipogenesis, that's fat production in the sebaceous glands in our skin.
The oil glands.
Exactly. So that suggests potential for treating acne by helping to balance out skin oil production.
Huh. Okay. And pharmacokinetics, how the body handles it. You mentioned something interesting there.
Right, PK is how the body absorbs, distributes, metabolizes, and excretes a drug. What was intriguing with CBC was that studies found high concentrations in both blood plasma and in brain tissue.
In the brain, too.
Yes. And even more, they observed accumulation after 14 days of repeated dosing.
Accumulation, so it builds up.
It seems so. It suggests CBC might be absorbed particularly well or maybe metabolized more slowly than some other cannabinoids. That buildup could be really important for how it's dosed therapeutically and how long its effects might last.
That is quite a profile for CBC. Okay, let's switch gears now to cannabinol, or CBN, often hyped as the sleepy cannabinoid. And I know this one's got an interesting backstory because the plant doesn't really make it directly.
That's absolutely right. CBN isn't a primary product from the plant's initial enzymatic steps like the others we've discussed. It's actually a degradation product.
Degradation. From what?
From A9-THC. Over time, especially with exposure to UV light and heat, THC slowly oxidizes and converts into CBN. So you often find higher concentrations of CBN in older cannabis that's maybe been stored for a while or exposed to the elements. That aging process is key to its formation. Think of it like THC getting mature.
So it's like an aged wine, but for cannabis. Okay, what about its pharmacology? How does it interact with our systems?
Well, its binding affinity, how strongly it sticks to both CB1 and CB2 receptors, is actually quite low, much lower than THC's affinity for CB1, for example.
So it doesn't hit the main cannabinoid receptors very hard.
Not really, no. But interestingly, it's quite active at those TRK channels again. It acts as an agonist, an activator at TRPV1, 2, 3, and 4, and also TRPA1.
Lots of TRP action.
Yeah. And uniquely, it acts as an antagonist, a blocker at TRPM8.
TRPM8. What does that one do?
TRPM8 is primarily involved in sensing cold and coolness, so blocking it might have some interesting sensory effects, though that's less explored. But the main point is, like CBG, CBN seems to work through multiple mechanisms, many of them outside the classic CB1, CB2 pathway.
Okay, let's get into its potential uses, starting with metabolic health again.
Similar to CBG, CBN shows orexigenic activity, meaning it stimulates appetite and increases food consumption in animal models.
Appetite stimulant again.
Yep. So, like CBG, it could be another potential non-narcotic, non-psychoactive agent for conditions like cachexia, where appetite loss is a problem.
Okay. And inflammation, does it play a role there too?
It does. CBN has demonstrated anti-inflammatory properties. One mechanism involves inhibiting the assembly of something called the NLRP3 inflammasome.
NLRP3.
Yeah.
That sounds important.
It is. It's a key part of the innate immune system's inflammatory response. By inhibiting its assembly, CBN can help dampen down excessive inflammation.
Gotcha.
It also affects other inflammatory signaling pathways, like inhibiting PNFEB and downregulating the pStat3-STAT3 ratio. And, like the others, it shows antibacterial efficacy against MRSA, another potential tool against resistant bacteria.
Okay, now we have to tackle the big claim. CBN is heavily marketed as the sleepy cannabinoid.
Oh.
What does the actual science say about its effects on sleep or sedation?
Right. This is where we need to be really careful and separate the marketing hype from the scientific evidence.
Okay.
While some early small studies and a lot of anecdotal reports link CBN to sedation, robust, controlled human studies specifically looking at CBN alone for sleep have produced. Well, let's call them equivocal or unsubstantiated results.
Not clear-cut.
Not at all, for CBN by itself. Now, there is evidence that CBN can increase the sedative effects when it's combined with demanded THC. That synergy seems to be real.
The combination effect again.
Exactly. But taking CBN in isolation as a sleep aid, the strong scientific backing just isn't there yet based on current human research. We really need to urge caution with that interpretation. It's not the guaranteed sleep potion some marketing suggests. Preclinical findings do suggest it might help reduce morphine withdrawal symptoms, which is interesting neurologically, but the sleep connection is tenuous on its own.
That's a really crucial distinction for listeners. It might enhance THC sedation, but isn't necessarily a standalone sleep aid.
Yeah.
And its pharmacokinetics. How does the body handle CBN?
It is detectable in brain tissue, especially at higher doses. But interestingly, unlike CBC, studies haven't found evidence of accumulation during repeated dosing.
No buildup over time.
Doesn't seem so. This suggests the body processes and eliminates CBN fairly efficiently without it stacking up, which could be relevant for safety and dosing strategies.
Right. Okay, shifting gears again, let's talk tetrahydrocannabivarin, or THCV. This one gets called a metabolic modulator. What makes THCV stand out? Let's start with its identity.
So THCV is one of those varinoids we mentioned earlier, derived from CBGVA, and it has that characteristic shorter 3-carbon propyl side chain.
The varin structure.
Exactly. It's usually found in smaller amounts in most cannabis strains, but certain varieties, often originating from Africa, can have higher levels. Its unique properties have definitely sparked a lot of scientific interest.
And its pharmacology sounds particularly intricate.
It really is quite nuanced. There are reports suggesting it can act as both an antagonist and an inverse agonist at CB1 receptors, depending on the dose and context.
Inverse agonist, what's that again?
Right, so an antagonist just blocks the receptor. An inverse agonist binds to the receptor and produces the opposite effect of a typical agonist. It actually reduces the receptor's baseline activity even when no agonist is present.
Wow, that's counterintuitive.
It is, and potentially very powerful therapeutically. THCV also shows partial agonist activity at CB2 receptors. And like the others, it interacts robustly with TRP channels, agonist at TRPV14 and TRPA1, antagonist at TRPM8, similar to CBN. Plus, it acts as a positive allosteric modulator of the 5-HT1A receptor.
Positive allosteric modulator. Meaning?
Meaning it doesn't directly activate that receptor itself, but it binds to a different site on the receptor and makes it easier for the body's natural signaling molecules, like serotonin, to activate it. It enhances the natural signal.
That's a lot of different ways it's interacting. Okay, let's dive into why it gets that metabolic modulator tag. This seems like where THCV really makes its mark.
This is definitely where THCV shines, yes. It shows a significant impact on metabolic health and research. Studies in obese mice found it helps regulate blood glucose levels, improves fasting plasma glucose, boosts glucose tolerance, and increases insulin sensitivity.
That all sounds very relevant for diabetes.
Extremely relevant. And importantly, a study in humans with type 2 diabetes found that THCV decreased their fasting plasma glucose levels.
Human data too, that's key.
Yes. Furthermore, some rodent studies showed it caused hypophagia, reduced food intake, and led to weight loss. The really exciting prospect here is its potential for appetite suppression and weight management, possibly without the kind of side effects seen with some older anti-obesity drugs. This makes it a really compelling candidate for tackling metabolic disorders.
That's incredibly promising, especially given the scale of issues like diabetes and obesity. What about its potential in the brain? Neurological or psychiatric uses?
THCV shows really diverse potential here too. For instance, in models of nicotine dependence, it was effective in reducing nicotine self-administration, inhibiting relapse behavior, and even preventing withdrawal symptoms.
Help with quitting smoking.
The potential seems to be there. In models related to schizophrenia, THCV reduced behaviors like hyperlocomotion and stereotypy, while actually increasing recognition, memory, and social interaction. So hitting multiple symptom types.
That's impressive.
In Parkinson's disease models, it reduced slow motor movements and crucially helped preserve dopaminergic neurons, the ones lost in Parkinson's. This suggests that it might have neuroprotective effects, potentially slowing disease progression.
Wow.
It also showed anti-epileptic properties, reducing seizure activity in animal models, and it acted as an antiemetic, reducing nausea effectively.
Gosh, that's an incredibly broad range of potential neurological benefits.
Yeah.
Addiction, psychosis models, neurodegeneration, epilepsy, nausea, and inflammation. How does THCV fit into that picture?
It has anti-inflammatory mechanisms too. It seems to mitigate signaling through the pan-X1-P2-X7 axis and suppress that NLRP3 inflammasome activation we discussed earlier.
Dampening the immune response again.
Exactly. It also downregulates the IL-6-TYK2-STAT3 pathway, suggesting, like CBC, it might have potential against cytokine storms. Plus, it reduces other inflammatory markers like PNF-K and pSTAT1. And interestingly, it also shows anti-acne effects, likely through regulating oil production and its anti-inflammatory action.
Okay, and finally, it's pharmacokinetics. How does THCV travel through the body, especially given all these effects?
This was quite interesting. One study found that THCV plasma levels were actually lower on day 14 of repeated dosing compared to day 1.
Lower over time. That's the opposite of CBC.
Right. It suggests that the body might adapt its metabolic pathways, becoming more efficient at breaking down and eliminating THCV with repeated exposure.
So maybe less risk of buildup, but potentially needing different dosing strategies.
Potentially, yes. It was detectable in the brain at higher doses. But again, no evidence of accumulation like with CBC. It seems to have a unique metabolic profile.
If you had to sum up the big picture for THCV, what really stands out?
I think it's that ability to hit such a wide array of targets relevant to both metabolic and neurological disorders, often without significant accumulation and, crucially, without the strong psychoactivity of THC. It really exemplifies that multi-target approach we see in the plant, hinting at maybe a more holistic way to treat complex conditions.
Fascinating. Okay, our final minor cannabinoid for today. Cannabidivarin, or CBDV. Sounds like a close relative of the famous CBD, right?
It is, yes. It's a structural homologue of CBD. Also a varinoid, so it has that shorter propyl side chain instead of CBD's pentyl chain.
Same varin structure as THCV then.
Exactly. And CBDV is often found in what are called landrace cannabis strains, basically. Older, naturally evolved varieties, often from regions like Central Asia or the Indian subcontinent.
Okay. What's its pharmacological signature? How does it interact with our systems?
Well, like CBD, CBDV has a pretty low binding affinity for both CB1 and CB2 receptors. It doesn't stick strongly to either. However, some studies suggest it might have greater potency or efficacy at the CD2 receptor compared to CB1, even if the binding is weak overall.
Okay. Slight preference for CB2, maybe?
Possibly, and it shares that similar TRP channel activity profile we've seen with others activating TRPV1, 2, 3, and 4 and TRP1 while inhibiting TRPM8.
The TRP connection again.
Yes. But what's particularly interesting with CBDV is its action as an inverse agonist at a receptor called GPR6.
GPR6. Okay, another one of those less common targets. Can you quickly break that down? What is GPR6 and why is interacting with it noteworthy?
Sure. GPR6 is what's known as an orphan receptor, meaning for a long time, scientists didn't know what naturally binds to it in the body.
Okay.
It's increasingly being recognized for its role in the brain, especially in areas involved in motor control, like the basal ganglia, and it's implicated in certain neurological conditions. So CBDV acting as an inverse agonist there, actively reducing the receptor's baseline activity, could be a really important mechanism behind some of the therapeutic effects observed for CBDV, especially in neurological disorders.
So even without strong action at CB1, CB2, it's hitting these other targets, particularly in the brain. Let's focus on that therapeutic potential, especially neurologically. What's the evidence?
CBDV has shown quite significant anti-epileptic properties. In lab studies using brain slices, it attenuates epileptiform activity, basically. It calms down the chaotic electrical storm associated with seizures.
It's almost a storm.
Yeah. And in animal models of epilepsy, it reduces seizure severity. There was also a human clinical trial focused on epilepsy.
Oh, human data again. What did it find?
It reported a 41% reduction in seizure frequency in the CBDV group, which sounds quite remarkable.
41%. That's huge.
It is. However, there was also a very high placebo response in that particular study, which made it difficult to show a statistically significant difference between CBDV and the placebo.
Ah, the placebo effect complicating things.
It often does in these trials, but it still points towards a potentially strong effect needing further investigation.
Absolutely. What about other neurological conditions?
This is really exciting. CBDV shows promise for symptoms related to autism spectrum disorder, ASD.
Really? How so?
In preclinical models, specifically models of Rett syndrome, which share some features with ASD, and also models using valproic acid exposure, CBDV was shown to ameliorate several behavioral abnormalities. Things like impaired sociability, repetitive behaviors, and memory deficits were improved.
Improving those core type symptoms.
Yes. in these models. And importantly, it seemed to have a favorable safety profile while doing so. This is a very active and hopeful area of research for these challenging neurodevelopmental conditions. Oh, and like THCV, it also showed antiemetic effects, reducing nausea.
That's incredible potential. Helping with ASD-related behaviors and epilepsy, potentially with good safety, is huge. What about inflammation outside the brain?
Yes, there's evidence there too. In animal models of ulcerative colitis and inflammatory bowel disease, CBDV showed positive effects on inflammatory markers. This effect seemed linked to its activation of the TRPA1 channel.
The TRPA1 channel again.
Yep. And in models of Duchenne muscular dystrophy, or DMD, a severe genetic muscle wasting disease driven by inflammation and muscle degeneration, CBDV was shown to improve muscle quality and locomotion, reduce inflammation markers, and actually help restore cellular repair processes called autophagy.
Wow, even in a complex genetic condition like DMD.
The preclinical data is certainly promising, suggesting it hits multiple relevant pathways: inflammation, muscle function, cellular repair. It really underscores its potential as a multi-target therapeutic.
Okay, as we sort of wrap up this deep dive, it's just abundantly clear, isn't it? These compounds we call minor are, well, they're anything but insignificant in their potential actions.
Absolutely not. Each one we've discussed has its own unique pharmacological fingerprint, interacting with the body in complex, diverse ways. They offer potential multi-target therapeutic approaches, often without the strong psychoactive effects of named THC.
Right.
The key takeaway for you listening is that these compounds might offer more nuanced, maybe more precise ways to influence our biology than just focusing on THC and CBD.
The big caveat.
The big caveat is that so much of this research is still preclinical. Animal studies, cell cultures, we desperately need more translational research and, crucially, more large-scale, well-controlled human clinical trials.
To bridge that gap.
Exactly. To confirm these effects in humans, figure out dosages, long-term safety, and really validate the therapeutic potential we're seeing hints of in the lab.
And what about that idea of the entourage effect? The notion that maybe these compounds work better together, synergistically, with each other, and maybe other cannabis components like terpenes?
Ah, the entourage effect. It's a really compelling hypothesis, isn't it? The idea that the whole plant, with all its diverse components working together as a team, might offer greater therapeutic benefits than just using isolated compounds.
Like a symphony versus a single instrument.
That's a good analogy. However, while the concept is popular and intuitively appealing, definitive, rigorous experimental data to fully prove specific synergistic interactions for most minor cannabinoids is still, well, it's still emerging. It's a very active area of research, and understanding those interactions could fundamentally change how we approach cannabis-based medicine in the future.
But we're not quite there yet with solid proof for everything.
Not for everything, no. And it's also really important for us to reiterate here. We're impartially reporting on the scientific findings from the sources provided. We're not endorsing any specific products or viewpoints, just trying to convey the information accurately.
Understood. Well, this has been a truly enlightening journey, hasn't it? Through this really intricate world of minor cannabinoids, from how they're born in the plant to their, frankly, remarkable potential, even if it's still emerging in areas like neurological disorders, inflammation, metabolic health. You listening now have a real shortcut to being well-informed about these compounds. They're definitely moving out of the shadows and into the scientific spotlight.
And it really leaves you wondering, doesn't it, as we keep uncovering these complexities in the natural world, what other compounds, maybe in cannabis, maybe in other plants, are just sitting there, holding undiscovered keys to our health, just waiting for someone to do a deep dive.
Yeah.
It's a powerful reminder that we've probably only just begun to scratch the surface of nature's pharmacy.
So true. Well, join us next time for another deep dive. We'll be exploring another fascinating part of the cannabis plant, the aromatic, often overlooked powerhouses, the terpenes.